So it's a great pleasure to be here today to present you the Metacell burden. It's a method to quantify the cell types liabilities on complex disorders when they are disrupted by rare variants. And today I'm going to show you an example of this method on the ASD, or autism spectrum disorder, and how we can identify the cortical cell types liabilities for this disorder. Let's start by defining what is autism. Autism, or ASD, it's one of the most studied or prevalent neurodevelopmental disorder, and it's characterized by two traits that appeared around the age of 3. So it's a problem with social interactions and stereotypical behaviors and restricted interest. And this disorder also have a lot of different other comorbidities, like we can see in this example of spiderweb plots. And this disorder also have a prevalence in the population around 3%. And we know that this disorder has a genetic factors explaining 50 to 80% of the different cases. But this disorder is also polygenic. So we cannot find only one variant on only one gene explaining the disorder. It's coming from different mutations in the different individuals. So we want to study how the genetic variants impact the risk for ASD. So we know that there is a relation between the risk of ASD and the variant frequency. So for example, we know that the severe risk variants are rare, and in contrast, the ASD-associated common variants have very small effect sizes on the disorder. And right now, most of our knowledge for the ASD risk derive from genes with de novo variants only. And previous studies found that 185 genes have de novo variants associated to ASD. And to understand the biological functions liability behind these variants, the authors try to find in which cell types these variants are enriched. And to do so, they used 20 different cortical cell types in the fetal times, and found that only one cell type was associated or enriched with ASD variants. And this cell type was an excitatory maturing neurons. However, we know that there is many other variants having an effect on ASD, but it's impossible to study these variants individually because they are partially inherited and therefore their effect sizes are smaller. So it's impossible, like I said, it's impossible to study them individually. But these variants still have an effect on ASD, and the function they alter is still unknown. So in this project, we have a huge knowledge gap, which is rare inherited gene disruptive variants increase ASD, but are still undocumented or understudied. And we know that they could explain the remaining unknown effect for ASD. So the aim of my work is to analyze aggregated rare variants or rare inherited variants to identify novel groups of gene-disrupted variants and corresponding cell types liabilities involved in ASD. And to do so, I've mixed two different studies, or not studies, but fields of research, which is a single-cell transcriptomic and the population genomics. Let's start by the transcriptomic. So we want to assign or to identify which genes are specific or assigned to a specific cell type. And to do so, we use the TDEP, so the top decile expression proportion, to define the specific genes to each cell types. And it assigns the top 10% most specific genes to each cell types and correspond to the genes with the highest proportion of their expression covered by the cell type. And with this method, each cell type will be defined or described by 1,675 genes. And To define the cell types we are going to work on and to assign the genes, we have used a public data sets of single nuclei RNA set made of 26 individuals and coming from prefrontal cortices at different ages. So we have individuals from the gestational week 22nd to the age of 40. And based on this article, the authors found that there is 18 different cell types based on the clustering and non-marker genes. So we have neuronal and non-neuronal cell types. And so we can assign the cell types to the 150,000 different cells composing this data set. However, these cell types were made from different ages, and we know that the genetic expressions vary a lot across the age. So we decided to split all the cell types in 6 different subclasses based on the age of the donor or of the individual. So, for example, here for the VIP inhibitory neurons, which is one type of inhibitory neuron, we have split it into 6 different classes. The fetal VIP neurons coming from fetal individuals to the adult VIP neuron coming from adult donors. And with this method, we assign each cell to a cell type and a developmental stage. However, we know that these combinations are still highly heterogeneous. So we decided to use not the annotation, but only the genetic profile to define some clusters of groups, some cluster of cells, which are called metacells. And we used 500, or we defined 500 metacells composed of around 300 cells per cluster to have enough cells to get a broad expression of the cell, of the metacell, and still have homogeneous expression within this group. And on this UMAP is represented the centroids of the 500 metacells, which are grouped on the different cluster we saw before. So if we use the same figures, we see that for each of the 150,000 cells, we assign a cell type, a developmental stage, and a metacell. And I used these 500 metacells to compute their most specific genes to identify which have the most effect on ASD. And to do so, I use a TDEP on the 500 metacells. The other part of my work was using human genetics. And the idea is to assign disrupted genes to each individuals, or genes disrupted by rare variants to each individuals. And to do so, we used 3 different cohorts, including around 65,000 individuals with a full sequencing, and with around 45% of them diagnosis of ASD. So for each individual, we have each individual will be defined by its different genes impacted by rare variants. So we have used 3 different kinds of variants: the genomic deletions, the genomic duplications, and the rare loss of functions (SNVs). And for the last one, it's made of 3 different SNVs: the stop gain, the frame shift, and the splice variant. And for example, here we can say that one individual is defined just by the sum of genes deleted. And the metacell burden I was presenting before, it's really the intersection between these 2 fields. So the idea is to study the impact of the genes associated to one cell type and to one individual when they are deleted in the individuals, and to compute the effect of this intersection of genes, or this overlapping genes, on a phenotype, and in our case is on ASD. So we used a logistic regression to compute the effect of each of these intersecting genes on the ASD. So I computed these effect sizes for the 500 metacells when the genes are deleted. So we can see if the genes specific to a certain metacells have an effect on the risk for ASD when it's deleted. And more a metacell or a centroid of metacell represented by these cycles is purple. Yeah, it's still purple on this screen. More it increases the risk for ASD. And if, I don't know if, yeah, you can see, if a metacell is cycled in black, it means that this effect is significant. And we can see that there is some clusters of effects of metacells with a significant effect sizes. And if we compare to the previous annotation found in the article, we see that, for example, we have some, I don't know if you can see my screen, my mouse. Yeah. Here we have some ID2 neurons, or ID2 neurons which have an effect on SD, which was already known in the literature. But we also found that oligodendrocytes have an effect, or genes specific to oligodendrocytes have an effect on ASD when they are deleted. And it was the same case for astrocytes, but it was never found before in the literature. And if we focus on the astrocyte, we see that we have only a subset of these astrocytes having an effect on ASD. And it's along the developmental stages. So it could be defining a new subtype of astrocytes only having an effect on ASD, while the rest of the astrocytes have no effects. But this representation is still a bit difficult to understand the effect or the liability of each cell types across time on their risk for ASD. So to do that, to have a better interpretation, we reassign the effect on each cell type. So we had the effect sizes for each metacells, and we reassign the effect size of each metacell to all the cells it was made of, and then we reassign the effect sizes to the annotations, which are cell types and developmental stages. And in this example, we can see that, for example, a fetal VIP is defined by its proportion of cells having a significant effect sizes on the risk for ASD. And each slice represents a group of cells annotated as fetal VIP and having significant effect sizes on ASD. And the color code is indicating the effect size. And we use this method to assign significant effect sizes for each cell types across the different developmental stages. And we found once again that the ID2 neurons, okay, it's impossible to see my mouse. The ID2 neurons have a huge proportion of significant effect sizes on the risk for ASD when their genes are deleted, when their specific genes are deleted. And with this representation, we also see that we have the L5-6 Themis neurons, deep layer excitatory neurons, having a huge effect on the risk for ASD, which was also something new in the literature. And for the astrocytes and oligodendrocytes we saw before, we see that it's only cells coming from later ages, or genes specific to cells coming from later ages having an effect on the risk for ASD. So we know that, for example, the ASD onset is around between infancy and childhood, and we see that the effect sizes appears at around the same time. And now that we understand a bit more the effect of deletion, we try to understand the other type of variants, duplications and the LOF. And we did the same analysis on the metacells. And an interesting thing is we found that, for example, for the oligodendrocytes, only the genes, the deletion of the specific genes and the LOF have an effect on the risk for ASD. but there is no effect at all for duplications. And once again, to have a better understanding of the effects of the cell types, we reassign the effect sizes to known annotations. And we have the proportion of significant effect sizes for the three types of duplication of variants: deletions, duplications, and the LOF. And for the oligodendrocytes, even if we saw that both deletions and LOF have a significant effect sizes, we saw that the deletions of the specific genes to oligodendrocytes have an effect only at later ages, while for LOF it was mostly at fetal time. So it showed that it's maybe different biological functions or biological functions having an effect on this disorder. And for both ID2 and L56 Themis neurons, we see that only their deletions of their specific genes have an effect on the risk for ASD. Both duplications and LOF have no effects at all. And yeah, sorry, I just forgot that I highlighted the thing. So yeah, and just to conclude, we could say that the burden analysis of the gene-disrupting rare variants specific to metacells identifies which cell types are the most associated to the risk for ASD. And we found that some consistency with the literature, like the deep layer excitatory neurons written L56 Themis neurons in this presentation. And we also identify new association with ASD, which was never seen before when the previous study focused only on de novo analysis. And we also saw that the cell type liability was highly dependent on the type of variant disrupting the genes associated to the cell types. So it's like not a conclusion, but maybe an opinion that in the future analysis focusing on rare variants, the type of variants impacting the genes should be always studied separately and not aggregated. And to conclude this presentation, we could say that integrating both single-cell transcriptomic and human genetics through the metacell burdens analysis can identify the cell type liability to the risk of ASD for any type of variants on any type of rare inherited variants, and it could be adapted to understand the association between any other complex traits and any other cell types if you want to focus on, I don't know, diabetes or other disorders. So I would like to thank both my labs, Dr. Jacquemon's lab and Noah Koski's lab at University of Montreal and UCSF, and of course all the different cohorts and patients coming from these three amazing cohorts. And thank you all for your attention. If you have any questions.
Any questions? So your logistic regression, you make a nice drawing, but I didn't see the formula, so I'm not sure exactly what is tested. Do you have like a cell effect and then a gene effect, or are you subsetting by cell and doing a burden test again for each metacell? I didn't get exactly which stuff.
So so for the logistic regression,
maybe you can just say what are you testing? Is it a model with an interaction term, or is it a series of logistic regression per metacell?
So yeah, we are doing one logistic regression per metacell, and on the y-axis is the ASD diagnosis, and on the x-axis we could say that it's the sum of genes both in the assigned to the metacell and to the individuals. So for each individual, we have the genes deleted or duplicated, depending on which variant we want to study, and also being assigned to the cell type we want to study. So it's really the sum of genes in the intersect we are studying. So yeah, thanks.
Thank you. Great talk. Just one quick question. I would like to understand why only 3 different variants selected, like deletions, duplications,
and LO
yeah,
and LOF.
yeah, and then the LOH. Why not gain of function or other types?
Which way?
Gain of
For the variants,
Yeah.
variants. Yeah, why not gain of functions and things like that?
We focus on these variants because it's known to be the variants having the most effect, and but we are starting to use and to include more variants like the missense and everything. But the issue is for missense variants, it's including a lot of different type of variants, and it's we need a lot of efforts to find if a variant is. We could use, for example, only the pathogenic missense variants, but we had to start somewhere, and we start by these variants. But in the future, yeah, we want to include more and more variants. Yeah.
Pathogenic is a good start. Thank you.
Yeah.